In topic 7.6, we'll be exploring the evidence of evolution. Evolution is an essential connecting theme in biology because it provides a unifying explanation for the diversity of life on Earth. It explains how organisms have changed over time and how they are related to one another. Evolution is the process by which populations of living organisms change over time through the mechanisms of natural selection, genetic drift, and gene flow. Evolution is important because it helps us understand the relationships between organisms, their physical and behavioral traits, and how they've adapted to different environments over time. It also explains the similarities and differences between species and why some species are more closely related than others. Additionally, evolution is critical for understanding the development of new species and the origin of life itself. By studying the process of evolution, biologists can learn how genetic variation arises, how new traits evolve, and how species diverge over time. Finally, understanding evolution is essential for many areas of biology, including ecology, genetics, and medicine. It helps us understand how organisms interact with each other and their environment, how diseases evolve and spread, and how to conserve and manage biodiversity. The evidence supporting evolution is diverse and robust. Since Charles Darwin's work over a century and a half ago, vast quantities of supporting evidence has been gathered and analyzed that would likely have amazed Darwin. More so, the types of evidence collected in recent times, especially those involving DNA and protein studies, as well as computer and mathematical modeling, has fortified our understanding of evolution and its steadfast position as the most important theme in all of biology. Rather than listing off hundreds of specific examples supporting evolution by natural selection ad nauseum, it's more appropriate to consider the types of evidence that those examples would be categorized into. To be sure, I will provide some focused examples to illustrate each of these categories. Morphological evidence includes the comparative anatomy and comparative embryology. Comparing the anatomy of different organisms can provide evidence for evolutionary relationships. For example, the forelimbs of vertebrates, such as humans, whales, bats, and birds, have similar structures but are adapted for different functions. Embryos of different species share similarities in their development, which can provide evidence for common ancestry. For example, all vertebrate embryos have gill slits and tails at some point during their development. Comparing the biochemistry of organisms provides evidence for evolution by showing similarities in the molecular structures of different organisms. Comparative biochemistry also reveals differences between organisms, which can be used to infer their evolutionary relationships. For example, the more similar the amino acid sequences of two proteins are, the more closely related the two species are likely to be. Conversely, if the sequences are very different, it suggests that the two species diverged from a common ancestor a long time ago. Geological evidence relies heavily on the fossil record and biogeography. Fossils provide a record of past life on Earth and show how organisms have changed over time. Biogeography studies the distribution of plants and animals across the world and can provide evidence for evolutionary relationships. It is important to note that these categories of evidence are not mutually exclusive, and evidence often can be categorized within more than one group. Think of them as occupying overlapping groups, as in a Venn diagram. Morphology is the branch of biology that deals with the form and structure of organisms, including their internal and external features. It includes the study of how organisms are put together, how they function, and how they've evolved over time. In morphology, scientists examine the physical characteristics of organisms, including their shape, size, color, and texture. They may also study the arrangement of organs and tissues within the body, as well as the function of specific structures and how they interact with the environment. Morphology is an important area of study in many fields of biology, including anatomy, zoology, botany, and paleontology. Homologous structures are physical structures in different organisms that have the same or similar anatomical features and evolutionary origins, even though they may serve different functions. These structures are evidence of shared ancestry and suggest that the organisms in question are closely related. Homologous structures can be found in a wide range of organisms, from mammals to insects to plants. Examples of homologous structures in mammals include the forelimbs of humans, cats, whales, and bats. While these animals use their forelimbs for different purposes, such as grasping, walking, swimming, or flying, their bones have similar structures and are arranged in similar ways. This suggests that the forelimbs of these animals have a common evolutionary origin. Homologous structures can also be found in plants. For example, the leaves of different plants may have the same basic structure, with a flattened blade, a network of veins, and a stem or petiole that attaches the leaf to the rest of the plant. While the leaves of different plants may have different shapes, sizes, and textures, they all share a common evolutionary origin and have similar underlying structures. A vestigial structure is a physical structure in an organism that no longer serves its original purpose, but is still present in the organism due to its evolutionary history. Vestigial structures are remnants of features that were once useful to the organism's ancestors, but have become unnecessary or maybe even detrimental over time. Examples of vestigial structures include the remnants of leg bones in some snakes, the reduced pelvic bones in whales, and the wings of flightless birds like penguins and ostriches. In many mammals, the ears can move independently of each other and can be used to detect sounds from different directions. This movement is controlled by muscles in the ears, which are not functional in the same way in humans. While humans do have those muscles, they are greatly reduced and therefore not capable of moving the ears in the same way as other mammals. Some people can still learn to control these muscles to some extent, but they really don't serve a useful function in terms of hearing or communication. Some vestigial structures are more physiological in nature. Goosebumps are the result of a physiological response when we experience strong emotions or when we're exposed to cold temperatures. Our body can respond by contracting the tiny muscles attached to the hair follicles on our skin, causing the hairs to stand up. In other mammals, the contraction of these muscles can cause the hair on the animal's body to stand up, which can help them to appear larger and more intimidating to potential predators or rivals. In humans, the absence of natural predators means this function no longer serves that purpose. For our early mammalian ancestors, the contraction of these muscles may have helped to provide insulation by raising the hair and trapping a layer of air next to the skin. But in humans, this reaction is no longer necessary for insulation, but remains as a vestige of our evolutionary history due to modern humans being significantly less hairy than our ancestors. Perhaps one of the most well-known examples of a vestigial structure in humans is the appendix. The appendix is considered a vestigial structure because it does not appear to serve any important function in humans, but is thought to be a remnant of our evolutionary history. In other mammals, such as herbivores, the appendix is much larger and thought to play a role in the digestion of plant material. However, in us, The appendix is much smaller and does not appear to have any significant digestive function. While the exact function of the appendix is not well understood, it is thought to be vestigial because it appears to be a remnant of our evolutionary past, when our ancestors may have needed a larger appendix to digest tough plant material. Over time, as humans evolved and our diet changed, the appendix became smaller and less functional, eventually becoming considered a vestigial structure. Although it is considered as such, recent research has suggested that it may play a role in the immune system, serving as a reservoir for beneficial bacteria and helping promote healthy gut flora. But the exact role of the appendix in the immune system is still not well understood, and many researchers still consider it to be, in fact, vestigial. Embryology is the study of the development of an organism from the fertilization of the egg to the formation of its body structures and organs. It is a branch of biology that focuses on the changes that occur during the embryonic stages of an organism's life cycle. Embryology plays an important role in understanding the evolutionary relationships between organisms, as similarities in embryonic development can provide evidence for common ancestry. For example, the similarities in the early embryonic development of vertebrates, such as the presence of a notochord, a cartilage-like structure that runs the length of the embryo in vertebrates and other close relatives, as well as slits or indentations of the pharynx of the embryo, suggest that all of these organisms in fact share a common ancestor. Comparative biochemistry is the study of the similarities and differences in the chemical processes and molecules that are involved in the metabolism, growth, and development of different organisms. It involves the analysis of biochemical pathways, enzymes, and molecules that are involved in various cellular processes and how they've evolved across different species. Comparative biochemistry also allows scientists to trace the evolution of particular biochemical pathways and enzymes. For example, the evolution of photosynthesis in plants can be traced by comparing the biochemical pathways and enzymes involved in photosynthesis across different groups of plants. Also, glycolysis is a fantastic example of a highly conserved process due to its ubiquitousness in nature. The same basic biochemical pathways are used by all living organisms to convert nutrients into energy, and many of the same enzymes and metabolic processes are used. The basic building blocks of life are the same across all living things, and the sequences of nucleotides in DNA and amino acids and proteins can be compared to show how closely related different species are. All living organisms use DNA as their genetic material, and the genetic code is nearly universal. Proteomics is the study of the structure, function, and interactions of proteins within an organism. It involves the identification, characterization, and quantification of the complete set of proteins, called a proteome, within a given biological sample. Comparing the amino acid sequences of proteins across different organisms can provide insight into their evolutionary relationships. Proteins that have a similar amino acid sequence in two or more organisms are likely to have evolved from a common ancestor. On the other hand, proteins that have distinct amino acid sequences in different organisms may have evolved through different evolutionary pathways. Proteomics can also be used to study the functional changes in proteins that have occurred during evolution. By comparing the proteomes of different organisms, researchers can identify proteins that have evolved to perform new functions or have evolved to change in their activity or regulation. These functional changes in proteins may have played a key role in the evolution of new traits and adaptations across different organisms. Even cellular structures can be considered evidence for evolution because they often show similarities across different species, and that suggests a shared common evolutionary history. All living cells contain a plasma membrane, which separates the cell from its environment, and a variety of organelles, such as mitochondria and the endoplasmic reticulum, that perform specific functions within the cell. One of the most compelling examples of cellular structures as evidence for evolution is the presence of mitochondria. Mitochondria are organelles that produce energy through the process of cellular respiration, as we know. We also know that they have their own DNA that is separate from the DNA in the cell's nucleus. The similarities between the DNA of mitochondria and that of bacteria suggest that mitochondria were once free-living bacteria and were engulfed by a larger cell, eventually evolving into a symbiotic relationship with that host cell. This process, which we know as endosymbiosis, is thought to have occurred early on in the evolution of eukaryotic cells and is one of the most well-supported scientific theories in evolutionary biology. And don't forget, chloroplasts share a similar evolutionary origin. Other examples of cellular structures that provide evidence for evolution includes the presence of flagella and cilia, which are used for movement in many different types of cells, and the similarity in the structure and function of the ribosomes that are responsible for protein synthesis in all living cells. Geological evidence is arguably some of the most important in studying the history of life on Earth. It includes the only connections available to our understanding of life in the distant and very distant past hundreds of millions of years ago: fossils. Considering that 99% of all species that have ever existed on Earth are now extinct, fossils are the only evidence we have that they were ever here. Fossils are the preserved remains or traces of organisms that lived in the past, and include things such as bones, teeth, shells, and footprints. They are important evidence for evolution because they provide a record of past life on Earth and show how organisms have changed over time. By studying fossils, researchers can reconstruct the evolutionary history of organisms and the processes that have driven their evolution. The fossil record provides evidence for the evolution of different groups of organisms over time, such as the transition from exclusively aquatic fish to partially land-dwelling amphibians and largely terrestrial reptiles, then to birds and mammals. They can also shed light on the evolution of specific traits, such as the development of feathers in dinosaurs and the evolution of the human brain. In addition to providing evidence of evolutionary change, fossils can also provide insight into the past environments and ecosystems in which organisms lived. By studying the fossil record, researchers can reconstruct the ecology and behavior of extinct organisms and how they interacted with other species and their environment. Biogeography as a subdiscipline is the study of the distribution of living organisms across geographic space and time, and the factors that have influenced their distribution. It seeks to explain why certain species are found in some places and not others, and how their distribution has changed over time. It is concerned with understanding the patterns of diversity and the presence of unique species found in specific geographic regions, as well as the processes that have led to the formation of these patterns. One of the key factors that influences biogeographic patterns is the history of Earth's continental movements and climate changes. Over millions of years, the continents have moved and collided, creating new land masses and changing the connections between different regions. One of the most well-known examples is the separation of the land masses that make up South America and Africa. These two continents were once connected as part of the supercontinent of Gondwana, but began to separate around 130 million years ago. As a result, many groups of organisms that were once distributed across both continents became separated, leading to the evolution of distinct lineages on each continent. The west coast of South America and the east coast of Africa are also interesting because they share similar geological features, including the presence of a deep oceanic trench and a chain of volcanoes. Another notable example is a group of flightless birds known as ratites, which include ostriches in Africa, emus in Australia, and rheas in South America. These birds are thought to have evolved from a common ancestor that was distributed across that supercontinent, but became separated as the continents drifted apart. Since time is a critically important factor in the study of evolution, dating of rocks and fossils make it possible to provide a timeline of history on Earth. Absolute fossil dating refers to the process of determining the age of fossil in years, typically through the use of radiometric dating techniques. Radiometric dating is based on the decay of radioactive isotopes, which occurs at a known rate into stable isotopes. By measuring the ratio of parent isotopes to daughter isotopes in a fossil or rock sample, scientists can calculate the age of the sample. There are several radiometric dating techniques that can be used to date fossils depending on the age of the sample and the isotopes present. Some of the commonly used methods include carbon-14 dating, potassium-argon dating, and uranium-lead dating. Carbon-14 dating is used to date organic materials up to about 50,000 years old by measuring the decay of carbon-14 isotopes in the sample. Potassium-argon dating is useful in dating rocks and minerals between 100,000 and several billion years old by measuring the decay of potassium-40 into argon-40 isotopes. Uranium-lead dating is used to date rocks and minerals that are billions of years old by measuring the decay of uranium isotopes into lead isotopes. By combining the results of different radiometric dating techniques, scientists can establish a timeline of the evolution of life on Earth, as well as the geological history of the planet itself. Relative dating is a technique used to determine the relative order of geological events and the age of rocks without necessarily determining their absolute age. It involves comparing the positions of rock layers in different locations, as well as the types of fossils found within those layers. Relative dating can be used to determine which rock layers are older or younger than others, and to establish a relative timeline of the geological history of a region. Relative dating relies on a number of scientific laws, and quite heavily on the law of superposition. This principle states that in a sequence of undisturbed rock layers, the oldest layers are at the bottom and the youngest layers are at the top. By using this and other principles of geology, we can determine the relative ages of rocks and infer the sequence of geological events that occurred in a region. For example, if a layer of rock containing a mammal fossil is found above a layer of rock containing a dinosaur fossil, then the mammal is presumed to be younger than the dinosaur. In many cases, observing evolution in real time is difficult. Evolution is a slow and gradual process that occurs over long periods of time, often taking thousands or millions of years to produce any significant change. As a result, it can be difficult to see the effects of evolutionary change, especially for organisms with long lifespans and slow reproductive rates. Additionally, evolution operates at the level of a population rather than individual, and changes in the frequency of traits or alleles within a population may not be immediately visible to the naked eye. Many evolutionary changes are driven by subtle genetic changes that may not produce obvious physical differences in the organism. For example, changes in the regulatory genes or non-coding DNA may have significant effects on an organism's development, behavior, or physiology, but might not be visible without specialized tools or techniques. In spite of challenges like these, scientific observation of evolution continues today. Quickly reproducing species, such as many insects, provide an excellent conduit for this evidence gathering since they can create many generations in a relatively short period of time. Perhaps the most well-known and very concerning example is that of the evolution of bacterial resistance to antibiotics. When bacteria are exposed to antibiotics, those that are resistant to the antibiotic are more likely to survive and reproduce, passing on their resistance genes to their offspring. Over time, this can lead to the evolution of bacterial populations that are highly resistant to multiple antibiotics. The antibiotic is the selective pressure that favors bacteria with resistance genes. Antibiotic resistance is a major public health concern, as it can make infections more difficult to treat and can increase the risk of complications and death. When antibiotics are overused, such as to treat minor infections that the immune system can fight off on its own given time and rest, or when they would be ineffective, such as for viral infections, or used in completely unnecessary ways, such as in cleaning products and hand soaps, this creates a scenario that favors the growth and spread of bacteria that are resistant to the antibiotics. To mitigate the risks associated with the over and/or improper use of antibiotics, it is important to use them only when necessary and to use them properly. This includes following appropriate prescribing guidelines, completing full courses of treatment, and avoiding their use for conditions that do not require them, such as the aforementioned viral infections. Another contemporary example is the emergence of the nearly 500 species of insects and other arthropods worldwide that have become resistant to pesticides. Like the bacteria example, when a pesticide is applied to a population of insects, those that are genetically resistant to the pesticide are more likely to survive and reproduce, passing on their resistance genes to their offspring. There are several mechanisms by which insects can develop resistance to pesticides. One common mechanism is called target site resistance, in which mutations occur in the genes that encode the proteins targeted by the pesticide. These mutations can reduce the affinity of the protein for the pesticide, making it less effective. Another mechanism is called metabolic resistance. In this case, insects evolve the ability to break down the pesticide more quickly, either through increased expression of detoxification enzymes or through mutation that enhance the activity of those enzymes. The evolution of pesticide resistance can have significant impacts on agriculture and public health. Resistance can reduce the effectiveness of pesticides, leading to increased crop damage, decreased yields, and increased reliance on more toxic or expensive pesticides. In addition, this resistance can make it more difficult to control disease vectors, such as mosquitoes, and other insect pests that have negative impacts on public health. The COVID-19 pandemic was caused by a novel coronavirus called SARS-CoV-2. One of the key features of this virus is the spike protein on its surface, which enables it to enter human cells. The spike protein is also the primary target of vaccines and antibody treatments. In the early stages of the pandemic, researchers noticed that there were two versions of the spike protein, D614 and G614, that differed by a single amino acid. Specifically, D614 had aspartic acid at amino acid position 614, while G614 had glycine at that position. Studies have shown that G614 is more infectious than D614. It is thought that this is because the G614 version is more stable than the D version, which allows it to bind more tightly to human cells and infect them more easily and replicate more quickly. From the virus's perspective, these are all clear evolutionary advantages. This is why the G614 version that emerged in Europe in early 2020 quickly became the dominant strain of the virus worldwide. And with that, this topic, and most important of all biology concepts, comes to a close. Thank you for watching, and take care.